Hello, my name is Jordan Wolf, and I reviewed the peer journal. It is Actin, Structure, and Function by Roberto Dominguez and Kenneth Holmes. In the journal, it says, under the control of hydrolysis, actin binding proteins and ions are able to transition between the G state, which is the monomeric, and F state, which is the filamentous. And it's the reason for actin being one of the main components in cellular functions being motility and the maintenance of the cell shape and the regulation of transcription and how polar it is. The interaction between F-actin, filamentous, and myosin forms the muscle contractions, and it owes its role to being the center role in the cell. The actin cytoskeleton is disrupted and taken over then by pathogens. Vertebrates express three main forms. They're called isoforms, and in the journal it talked about skeletal, cardiac, and smooth muscle isoforms, and there is a beta and a Y as well, but those are expressed in non-muscle and muscle cells. Further, they talked about actin isoforms, and they differ from the skeletal, cardiac, and smooth because they only have a few amino acids, and most of the variations occur towards the N-terminus, which is down to the left of the first domain. Nucleotide hydrolysis by F-actin is one of the main factors between G and F-actin, and the ATP state is more stable than the ADP state. The actin monomers joined at the positive end, which is called the barbed end, and the end of the filament is in the ATP state. Hydrolysis takes place in the filament, and ADP actin monomers dissociate from the negative end. The steady state mechanism of actin polymerization and depolymerization is known as actin filament treadmilling, and they talked a lot about treadmilling in the article. And further down, they talked about G-actin binding proteins. They went on to talk about how they have diverse functions and belong to different structural families, and are present in different nucleotide preferences. Despite the diversity, most of the proteins bound in the target binding cleft of actin and typically insert an amphibulic helix into the cleft. And thank you.